Welcome to the deep dive. Have you ever just, stopped to marvel at the sheer mind-boggling complexity of life?
It's pretty incredible when you think about it.
Yeah. I mean, think about it. Every single cell in your body, from a nerve cell firing off signals to a muscle cell doing the heavy lifting, has the exact same genetic instructions,
The exact same blueprint.
but they look and function so differently. It's like having one instruction manual, but using it to build both a spaceship and, I don't know, a sailboat.
Right. How is that even possible? Well, that's the magic, or rather the science, of gene regulation.
Oh.
It's this really ingenious system that controls when and how much of each gene gets expressed, turning genes on, off, dialing the volume up or down.
Creating all that diversity from the same starting point.
Precisely, and allowing cells to adapt and develop and maintain their specific jobs.
So that's our mission today then, to sort of peel back the layers on that control system in eukaryotes.
Exactly. We're talking protists, fungi, plants, animals, us included. We want to unpack the mechanisms they use.
And our guide for this is a chapter from Brooker's Genetics, Analysis and Principles, 7th edition.
Yeah, that's the one. A really solid resource. We'll be pulling out the key ideas on transcriptional and translational regulation. So how genes are read and how the messages are used.
And this isn't just abstract science, is it? Understanding this helps explain, well, everyday biology.
Absolutely. It's fundamental to everything from, like you said, getting a tan to how your body manages iron, even down to the roots of certain diseases.
You get to see just how incredibly precise these cellular controls really are. Okay, let's dive in. Where do we start?
Let's start with the main conductors, you could say, the regulatory transcription factors.
Okay, transcription factors. I know there are general ones that RNA polymerase needs just to start transcribing any gene.
Right, this is the basic machinery. But the regulatory ones are different. They're the proteins that fine-tune the rate of transcription for specific genes. They're the volume controls.
And they don't just bind anywhere, do they? They recognize specific DNA sequences like little docking stations?
Exactly like docking stations. We call these sequences cis-acting elements or regulatory elements, usually found near the gene's main starting point, the core promoter.
And these regulatory factors, they can either boost things up or tamp things down.
Precisely. They can be activators, which bind to DNA sequences called enhancers. And when they do, they can seriously ramp up transcription. We're talking 10, 100, even 1,000 times more.
Wow, 1,000-fold. That's huge.
It is. Or they can be repressors. These bind to different sequences called silencers, and they do the opposite. They prevent or decrease transcription. That's downregulation.
Okay, enhancers boost, silencers block. But you mentioned these elements aren't always right next to the gene.
No, and that's one of the really fascinating and complicating things. They can be thousands, even hundreds of thousands of base pairs away.
100,000? How does that even work?
DNA looping. The DNA can actually bend around so that an enhancer or silencer far away physically comes close to the promoter region.
Ah, okay. So distance isn't necessarily a barrier.
Not at all. And they can often work in either orientation, forward or backward, and sometimes even be located within a gene in those non-coding intron regions.
Which must make finding them incredibly difficult for researchers.
Oh, absolutely. It complicates things significantly. And it points to another really key concept, especially in multicellular organisms: combinatorial control.
Combinatorial, meaning it's not just one factor controlling one gene.
Right. It's usually a combination of factors. You might have several different activators, maybe a repressor or two, all binding in the vicinity. Their collective effect determines the final level of gene expression.
Plus, you can have things modifying the activators or repressors themselves, or changes to the DNA packaging.
Exactly. Or DNA methylation, which we'll get to. It's a whole committee making the decision, not just one boss.
So how do these factors physically interact with the DNA? What allows them to recognize and bind those specific enhancer or silencer sequences?
They have specific 3D shapes. These proteins have distinct regions called domains. One domain might be responsible for binding to the DNA. Another might bind to a signaling molecule, for example.
And within these domains, there are common structural patterns.
Yes, called motifs. These are like recurring architectural designs found in many different transcription factors.
Like the alpha helix. I remember that one fitting neatly into the major groove of the DNA double helix.
That's a classic one, the recognition helix. It uses hydrogen bonds, and its positive charge interacts nicely with the DNA's negative backbone.
And there are others like helix-turn helix, helix-loop helix.
Zinc fingers, too. Those use a zinc ion to stabilize the structure. They all have ways of reading the DNA sequence in the major groove.
And I remember the leucine zipper being interesting because it helps two transcription factor proteins come together.
Right, it promotes dimerization. You can get homodimers, two identical factors joining up, or heterodimers, where two different factors pair.
Which adds another layer of complexity and potential control, right?
Yeah.
Different pairings could have different effects.
Absolutely. More combinations are possible. Now, these factors usually don't interact directly with the RNA polymerase enzyme itself. They work through intermediaries.
Okay, so who are the go-betweens?
One major player is a complex called TFIID. Activators might help TFIID bind better to the promoter, essentially recruiting it. Repressors might block TFIID.
And there's another one, mediator.
Yes, the mediator complex. It's a large protein complex that, well, mediates the interaction between the regulatory factors and RNA polymerase II.
How does it do that?
Activators can stimulate mediator to add a phosphate group phosphorylate, a part of RNA polymerase II called the CTD. This phosphorylation is like a green light, allowing the polymerase to move from initiation to actually elongating the RNA strand.
And repressors would presumably prevent that phosphorylation, put the brakes on.
Exactly. They inhibit mediator's kinase activity or block its interaction.
So even the transcription factors themselves need to be controlled. They aren't just always on.
Definitely not. Their activity is tightly regulated to ensure genes are expressed only when and where needed.
And you mentioned three main ways this happens.
Yeah, three common mechanisms. First, the binding of small effector molecules, like hormones, can directly switch a transcription factor on or off.
Second, protein-protein interactions, like the dimerization we talked about. Forming a dimer might be required for activity.
And third, covalent modifications, like adding a phosphate group phosphorylation or other chemical tags.
Let's look at an example. How do steroid hormones work, like glucocorticoids?
Great example. Steroid hormones are small lipids. They can easily pass through the cell membrane. Inside the cell, a glucocorticoid hormone binds to its specific receptor protein, the glucocorticoid receptor.
And that binding does something.
It causes a chaperone protein called HSP90 to detach from the receptor. This uncovers a hidden signal on the receptor, a nuclear localization signal, NLS.
Ah, the NLS tells the cell, take this into the nucleus.
Precisely. Then two of these activated receptors pair up, form a homodimer, travel into the nucleus, and bind to specific DNA sequences called glucocorticoid response elements, or GREs.
And these GREs are located near the genes that the hormone needs to activate.
Exactly. The binding of the receptor dimer to the GRE then recruits coactivators and stimulates transcription of those target genes. Things involved in, say, glucose synthesis or fat mobilization.
So one hormone signal fans out to activate a whole set of related genes, even if they're far apart on the chromosomes.
It's a coordinated response, similar in concept to bacterial operons, but much more spread out.
Okay, what about that covalent modification example?
Yeah.
The CRA protein.
Right, the CREB protein, CAN-MP response element binding protein. This one is regulated by phosphorylation.
So something happens outside the cell first.
Usually, yes. An external signal, like a hormone binding to a receptor on the cell surface, triggers a chain reaction inside. This often involves producing a second messenger molecule called CAMP.
And CAMP activates something else.
It activates protein kinase A. This enzyme then travels into the nucleus and adds a phosphate group to the CRE protein. It phosphorylates it.
Now, here's the clever part I remember. CREB can bind to the DNA whether it's phosphorylated or not, right?
Correct. It binds to a specific DNA sequence called the CAM-MP response element, CRE.
But only the phosphorylated CREB can then bind to another protein, a coactivator called CBP.
Exactly. It's that binding to the coactivator, CBP, that's necessary to actually activate transcription. The phosphorylation is like a license to recruit the final activating machinery.
Wow. So many layers of control just to turn on a gene. It's amazing. It works so reliably.
It really is. The precision is incredible.
Okay, so we've talked about the factors that read the DNA, but what about the DNA itself? It's not just a naked strand floating around. It's packaged up, right, into chromatin.
That's the next crucial piece. DNA is wrapped around histone proteins, forming structures called nucleosomes. This packaging is essential to fit our enormous genome into the nucleus, but it also presents a barrier.
Yeah, how does RNA polymerase read DNA that's tightly wound up? It seems like it would be inaccessible.
It often is. We talk about closed conformation chromatin, which is tightly packed and hard to transcribe, versus open conformation, which is looser and more accessible.
So how does the cell switch between these states?
Through ATP-dependent chromatin remodeling, this involves multi-protein machines, these complexes, that use the energy from ATP hydrolysis to physically alter chromatin structure.
What can they actually do?
Three main things. One, they can change the position of nucleosomes, slide them along the DNA, or change the spacing between them.
Okay, making space.
Two, they can actually evict the histone octangers,
Yeah.
the core histone proteins creating gaps, regions free of nucleosomes.
Literally kick them out.
Temporarily,
Yeah.
yes. And three, they can replace standard histones with histone variants.
Histone variants. So not all histones are identical.
No. While most histone genes make the standard proteins, there are slightly mutated versions. Variants exist for H1, H2A, H2B, and H3, but interestingly, not really for H4, which is highly conserved.
And these variants have different jobs.
They do. Some, like H2AZ or H3.3, are often found near active genes and seem to create a more open or easily remodeled state. Others might be involved in repression, like macro H2A on inactivated X chromosomes, or specialized functions like SENH3 at centromeres for chromosome segregation.
So the type of histone present matters. What about modifications to the histones?
Ah, yes. That leads us to the histone code hypothesis. The tails of the histone proteins stick out from the nucleosome core.
Right, those N-terminal tails.
And these tails can be chemically modified in dozens of ways: acetylation, methylation, phosphorylation, ubiquitination. There are over 50 enzymes known to do this.
And the code idea is that the pattern of these modifications carries information.
Exactly. It suggests that specific combinations of modifications act like signals, a language read by other proteins.
Like acetylation. Adding an acetyl group to lysine residues neutralizes their positive charge.
Right. And since DNA is negatively charged, this loosens the histone's grip on the DNA, generally promoting a more open chromatin state favorable for transcription.
Other modifications might attract proteins that condense the chromatin, or recruit those remodeling complexes we mentioned.
Precisely. The pattern tells other proteins whether to activate or repress that region of the genome.
Okay, this is getting complex. How do scientists figure out where all these nucleosomes are and which modifications they have across the entire genome?
That's where a powerful technique called ChIP-seq comes in, chromatin immunoprecipitation sequencing.
ChIPs. Sounds technical. How does it work?
Well, first you treat living cells with a chemical, like formaldehyde, that cross-links proteins to the DNA they're bound to, freezes everything in place.
Okay, locks the histones onto the DNA.
Then you break the DNA into smaller fragments. Crucially, the DNA wrapped around histones is protected, while the linker DNA between nucleosomes gets digested.
So you end up with DNA fragments mostly corresponding to the length wrapped around one nucleosome.
About 150 base pairs, yeah. Then comes the IP part, immunoprecipitation. You use antibodies that specifically recognize and bind to the protein you're interested in.
So an antibody for a specific histone modification, like acetylated H3, or a specific histone variant.
Exactly. These antibodies are attached to tiny beads. You mix them with your chromatin fragments, and the antibody fishes out only the nucleosomes carrying that specific modification or variant.
Like molecular fishing.
Pretty much. You purify these captured nucleosome DNA complexes, remove the cross-links and the proteins, and you're left with the DNA fragments that were associated with your target histone feature.
And then you sequence that DNA, the sick part.
Right. High-throughput sequencing tells you the sequence of all those fragments. Then you use computers to map those sequences back to the reference genome,
allowing you to see exactly where in the genome that specific histone modification or variant was located.
Precisely. You get a genome-wide map. And ChIP-seq studies revealed something quite common. Many active gene promoters have a nucleosome-free region, NFR, right at the start.
An open space, about 150 base pairs wide.
Typically, yes. Flanked by well-positioned nucleosomes, often containing variants like H2AZ that are easier to move. This NFR acts like a landing pad for the transcription machinery.
Okay, let's put it all together. How does a gene actually get turned on, considering all these chromatin factors?
It often starts with an activator transcription factor binding to its enhancer sequence, maybe within one of those NFRs.
Step 1, activator binds.
Step 2, That activator then recruits helpers, typically a chromatin remodeling complex like Swiss NF, and a histone-modifying enzyme like a histone acetyltransferase, HAT.
Okay, recruits the remodelers and modifiers.
Step 3. The remodelers use ATP to slide or evict nucleosomes around the promoter and the start site of the gene. The HAT acetylates histones, loosening their grip on the DNA. This opens up the region.
Creating access for the main machinery.
Exactly. Step 4. Now, the general transcription factors and RNA polymerase II can bind to the core promoter, forming the pre-initiation complex.
And transcription begins. What about as the polymerase moves along the gene?
Good question. Step 5. During elongation, histones ahead of the moving polymerase often get further modified, like acetylation, and evicted, perhaps handed off temporarily to histone chaperones.
So the path is cleared.
Yes. And step 6. Behind the polymerase, the histones are put back in place, often deacetylated, restoring the chromatin structure. This helps prevent spurious transcription from starting within the gene. Histone variants like H3.3 also play roles here.
It's a highly dynamic process. Unpack, read, repack.
Very dynamic, constantly changing.
Okay, one more major layer of regulation. DNA methylation. This isn't modifying the histones, but the DNA itself.
Correct. It's the addition of a methyl group directly onto a cytosine base, usually when it's followed by a guanine, the CpG dinucleotide. An enzyme called DNA methyltransferase does this.
And you said this is common in vertebrates and plants.
Very common, yes. Less so in insects like Drosophila or in yeast.
What's the effect of methylation?
Generally, it acts as a silencing signal, especially when it occurs in dense clusters called CpG islands near gene promoters.
CpG islands.
Huh.
So regions with lots of CpGs.
Right. For housekeeping genes, the ones needed in almost all cells, their CpG islands are usually unmethylated, allowing them to be expressed.
But for genes that should only be active in specific tissues.
For those tissue-specific genes, methylating their CpG islands in other tissues is a key way to keep them switched off.
How does the methylation actually block transcription?
2 main ways. First, the methyl group itself can physically block the binding site for some activator transcription factors. They just can't latch on.
Okay, direct interference.
Second, there are proteins called methyl CpG binding proteins that specifically recognize methylated CpGs. When they bind, they recruit other proteins, like histone deacetylases, HDACs.
So they bring in enzymes that remove those activating acetyl marks from histones.
Exactly, which helps compact the chromatin into that closed, transcriptionally silent state. So methylation leads to histone deacetylation and silencing.
And these methylation patterns can be passed down when cells divide.
Yes, that's crucial. There's maintenance methylation. After DNA replication, the old strand is methylated, but the newly made strand isn't. Special DNA methyltransferases recognize this hemimethylated state and copy the pattern onto the new strand.
So the pattern is preserved, maintaining cell identity through divisions.
Precisely. It's also fundamental to processes like genomic imprinting, where methylation marks laid down during egg or sperm formation determine which parent's allele is expressed in the offspring.
This complexity is just astounding. Which brings us to that huge project, encode, the encyclopedia of DNA elements.
Right. A massive international effort launched in the early 2000s. The goal was ambitious. Identify all the functional elements in the human genome, not just the genes, but all the regulatory switches, the enhancers, silencers, promoters, insulators, everything.
How do they even attempt that?
They used a whole suite of techniques, including ChIP-seq for histone modifications and transcription factor binding sites, methods to map DNA methylation, RNA sequencing to see what's actually transcribed, mapping regions sensitive to DNase I enzyme, which cuts open chromatin.
A coordinated onslaught of molecular mapping.
Pretty much. And the initial results, published around 2012, were stunning.
What did they find?
They concluded that something like 80% of the human genome sequence is linked to some biochemical function.
80%. That flew in the face of the old junk DNA idea, didn't it?
Completely. It suggested that the regulatory landscape was far vaster and more complex than previously thought. They identified millions of potential regulatory regions.
What are the implications of that?
Well, it highlights that gene regulation is incredibly intricate, influenced by vast networks of elements near and far. And crucially, it has big implications for understanding disease.
How so?
Many genetic variations identified through genome-wide association studies, GWAS, as being linked to common diseases like diabetes, heart disease, autoimmune disorders don't fall within protein-coding genes.
They fall in these non-coding regulatory regions.
Exactly. Encode provided a map to start understanding what these non-coding variants might actually be doing, perhaps disrupting an enhancer, creating a new transcription factor binding site, altering methylation.
So it's a vital tool for deciphering the functional consequences of genetic variation related to health and disease.
Absolutely. It's giving us clues about how regulatory disruptions contribute to disease pathology.
Okay, we've covered a lot on controlling transcription. But regulation doesn't stop once the mRNA is made, does it?
Not at all. There's a whole other level of control operating on the mRNA itself, regulating its translation into protein, or controlling its stability and degradation. This is post-transcriptional regulation.
The final checkpoints. Can we look at an example?
A really elegant one is how our cells manage iron assimilation. Iron is essential, but too much is toxic.
So there needs to be tight control over uptake and storage.
Precisely. Cells take up iron bound to a protein called transferrin, using the transferrin receptor on the cell surface. Excess iron gets stored safely inside a protein complex called ferritin.
And the regulation involves an RNA-binding protein.
Yes, the key player is the iron regulatory protein, IRP. It can bind to specific stem loop structures in certain mRNAs called iron response elements, IREs.
Okay, IRP binds to IREs. Where are these IREs found?
This is the clever part. In the mRNA for ferritin, the storage protein, the IRE is located in the 5U untranslated region, 5UUTR. That's the part before the protein coding sequence starts.
So what happens when IRP binds there?
When iron levels in the cell are low, IRP binds tightly to this 5UUTR IRE, and this binding physically blocks the ribosome from initiating translation.
So low iron means no ferritin gets made. Makes sense. You don't need to store iron if you don't have much.
Exactly. But when iron levels are high, iron atoms bind directly to the IRP. This causes IRP to change shape and release the IRE.
Freeing up the mRNA.
Right. Now, the ribosome can bind and translate the ferritin mRNA, producing more ferritin protein to safely store the excess iron.
A beautiful feedback loop right at the translation level. What about the transferrin receptor, the protein that brings iron into the cell?
For the transferrin receptor mRNA, the IRE is in a different place. It's located in the 3-oro untranslated region, 3-oro UTR, the part after the protein coding sequence stops.
Okay, at the other end, does IRP binding there also block translation?
No, this is the fascinating twist. When iron is low, IRP binds to this 3-oro UTR IRE, but instead of blocking translation, it stabilizes the mRNA. It protects it from being degraded by cellular enzymes, endonucleases.
So low iron leads to more stable transferrin receptor mRNA.
Yes, which means more transferrin receptor protein gets made, increasing the cell's ability to grab whatever scarce iron is available outside.
And when iron is high?
When iron is high, iron binds IRP, causing it to release the 3-oro UTR IRE. Now that protective binding is gone, the transferrin receptor mRNA becomes unstable and gets rapidly degraded.
So less receptor is made, reducing further iron uptake. It's perfectly coordinated.
It's incredibly elegant. The same protein, IRP, acting as a sensor, regulates two different mRNAs in opposite ways, just based on where the IRE sequence is located in the mRNA, all to maintain iron homeostasis.
That really shows the power and subtlety of post-transcriptional control.
Wow, it's a fantastic example.
Okay, we have covered an incredible amount of ground today. We've gone from the regulatory transcription factors acting as conductors, through the complex packaging of chromatin and histone modifications, the silencing effect of DNA methylation, the huge mapping effort of encode, and finally down to the fine-tuning of translation with the iron example.
It really paints a picture of layers upon layers of control, all working together to ensure the right genes are active at the right levels, at the right times, in the right cells.
Building and maintaining the sheer complexity of eukaryotic life.
It's truly remarkable how dynamic and fluid gene expression actually is. All these enhancers, silencers, chromatin states, methylation patterns.
It makes you think, doesn't it? Given how much the expression of a gene depends on all this surrounding context, the cell type, the environment, the modifications, what does that tell us about how we even define a gene in complex organisms? Its function seems so contingent on its regulation.
That's a deep question. It certainly challenges a simple static view of what a gene is. The regulatory information is arguably as important as the coding sequence itself.
Something definitely worth mulling over. It gives you a lot to think about.
Absolutely.
Well, thank you for joining us on this deep dive. We hope exploring these intricate mechanisms has given you a new appreciation for the elegance of cellular control. Thank you for being part of the Deep Dive family.
Thanks for listening.
We'll see you next time.